Hello everyone. So in today's video, we are discussing the question from GATE Life Sciences 2015 paper. So this question I have taken from the biochemistry portion. So in this question, you can see a table is being provided and the four different proteins are given. Their molecular weight in the native form is given. Their PI values are given and their type is given. So it has been asked that which one of the following options correctly identifies the order of elution in size exclusion chromatography and the increasing order of mobility in SDS polyacrylamide gel. Okay, so here it has been talked about two techniques: size exclusion chromatography and the SDS polyacrylamide gel electrophoresis. Okay, so let's talk about these first and then we'll get to the question. Okay, so uh, chromatography, which is a technique to separate the biomolecules. Okay, so we have different types of chromatography. Okay, so the major types of chromatography I'm writing. One is TLC, or we can also say thin layer chromatography. Another is the column chromatography. Have affinity chromatography. We have gel filtration or size exclusion chromatography. We have ion exchange chromatography. So these first chromatography is the absorption based, and the where you can separate the biomolecules based on their adsorption on the silica membrane. So there we have a thin layer of glass coated with silica. This silica which is polar in nature. So there you spot your mixture of samples and put it in a beaker which has mobile phase. Okay, so in all the chromatography we have two phases. One is the stationary phase, another is the mobile phase. So here silica is the stationary phase, which is a solid, and the liquid, which is a mobile phase, which is a solvent. Okay, so if we are using a non-polar solvent and our silica which is polar, so when the mixture, the components, they'll start separating, they will adsorb to the silica based on their polarity. So if the polarity, the compound is polar, it will make affinity, it will have high affinity towards silica, and it will, so once the liquid start when we dip the paper in the liquid, and we dip the paper in the liquid, so the liquid starts getting absorbed to the paper and it moves upwards in the upward direction via capillary action. So when so the components will start separating. So the component which will have the high polarity which will have the high polarity, it will have more affinity towards the silica. Won't be able to migrate much. But the one which is having the which is not polar and have less affinity towards the silica, it will keep moving upwards towards with the mobile phase, because it will have more affinity towards the mobile phase, which is also non-polar. So that is how in the TLC, thin layer chromatography, we separate the molecules via their polarity. Similar, we do column chromatography also, but here we don't use uh, here the solvent, we don't put it in the bottom so that it reaches upwards towards the capillary action. Here we make a silica column, a glass packed with silica in that column, and there we put our solvent or mobile phase from the upward, and the mixture will get eluted according to the polarity. So here also the non-polar will come first, and then the polar one comes later, because it will have more polarity with the stationary phase, that is the silica. Next, we have the affinity chromatography. So in affinity chromatography, we purify certain proteins using the affinity towards the column or the beads. Very common example is the nickel anti affinity purification. Next, we have this gel filtration and size, or we can say size exclusion chromatography. So using size exclusion, we can separate the biomolecules based on their sizes. So if we have uh, a mixture of protein, one which is of higher size, one which is of lower size. Okay. This is the ladder. So, okay. So first, if we have one mixture, okay, mixture of protein, you run it on SDS-PAGE. So SDS-PAGE, what is this? It is a reducing page. So SDS reduces or breaks the protein into its primary structure, and in the primary structure, and when we ran it on the SDS-PAGE, the molecules start migrating. There also separation happens, so they will migrate towards the positive charge. As the SDS is an anionic detergent, it coats the all the proteins. All the proteins were coated with SDS, they have the negative charge, and they will migrate from negative to positive uh, movement in the electrophoresis system. So there also, even though all the proteins will move towards the positive charge, but in the polyacrylamide gel, the one which is having the lower molecular weight will move faster compared to the higher molecular weight. Okay, so that is how the movement take place in SDS-PAGE electrophoresis system. Okay, but if you have impurities here, or if you have a mixture of proteins here and you want to separate the larger and the smaller proteins from each other, what you can do, you can perform the size exclusion chromatography, where the column is packed with the porous beads. Okay, these porous beads, and here you put the bigger proteins, and the smaller one. Okay, so the protein which is smaller in size, it can enter the pore, but the larger one cannot enter the pore. Okay, huge large. Okay, so the larger protein molecules cannot enter the pore, so they will uh, cross this column from the void. They will be using the void volume. As they are coming through the void, it's an easier path for them to cross, and these higher molecular weight will come out or will elute out from the column first. Okay, these will be eluted out first, but the smaller one, they are entering to the pore, so they will uh, pass through the pores, and that's why they have a larger path to cover. So in the second elute or in the later elutes, the smaller proteins will come out. So that's how you can separate the smaller and bigger proteins from each other using the size exclusion chromatography. Okay. So next one is the ion exchange chromatography. Here you can purify. So previously you have purified using the sizes of the protein, but in ion exchange, if even though the sizes are same, you can purify the protein based on their charges. So if your protein of interest, suppose it has a positive charge and you want to purify, you can use cation exchange. Okay. So here also you have a immobilized surface, charged surface. Okay. So there this so this is the example I'm giving of anion exchange. Okay. So this is a immobilized surface where uh, loosely attached buffer ions are present, but these are the immobilized cations. When you put a protein mixture, okay, protein mixture to it, so what it does, these anions which is have higher affinity will exchange these buffer anions, and it will bind to the immobilized surface. It will bind to the immobilized surface, and those other non-specific one will be come out. Okay, and it will remain, the negatively charged protein will be bound to the immobilized cationic surface. And when you provide a high salt concentration, so that high salt replaces this bound anions, and then in that high salt concentration scenario, these proteins will come out of the column. So first all the non-bound one will come out, and then when you increase the high salt concentration, high salt when you increase the salt concentration, the in for the elution of the bound protein, the bound negatively charged protein will be eluted out, and that's how it will separate out based on the charges. Okay, so that is the ion exchange chromatography. So in the question, so it has been given the information of molecular weight, pi and type. So here they have performed size exclusion chromatography. It has been asked, correctly identify the order of elution in size exclusion chromatography. So first let's see size exclusion. So order of elution. So which one will come first and which will come later. So the first, which the protein will come out which will be having the higher molecular weight. So if we'll see, so in size exclusion chromatography we are not treating it with any detergent. So the native structure of the protein remains intact. So if you'll see the sizes, it so the S protein has the higher size, that is 45 kilodalton. So and then uh, Q protein which is 40 kilodalton, P protein which is 32 kilodalton, and R protein which is 45 kilodalton. So that is the order of the elution, because the higher molecular weight will come out or will be eluted first. So S followed by Q followed by P and followed by R. So S, Q, P, R. So here option D you can see in the chromatography, this is the order of elution via size exclusion chromatography. Now if the same condition is protein mixture is run on the SDS page. So as the SDS page is a reducing page, it provides it makes them in a primary structure. Okay, so As you can see, the first protein P, which is a monomer. So monomer, that means its size will be 32 kilodalton only in the native page. Second one Q, which is a homodimer. So in the SDS page, it will be separated into its dimer. The dimer will be separated and it will create a single site. It is a homodimer, that means both are same protein subunits. So individually 20 kilodalton. The R protein is also monomer only, so it will remain in its native size. The third one, which is a homotrimer, that means similar subunit 3 copies, that means individually 15 kilodalton. So all three will migrate at the same level at this according to the size, that is the 15 kilodalton. So there it has been asked in the increasing order of mobility. So the lower mobility will be of higher size of the protein. So now if we'll see the higher size, so in all of them in the native form the highest size was S, but in the reduced form the highest size is of P, and then R, then Q, and then S. Okay, P, R, Q, S. So in the native page, the migration will be, so the lowest migration is of 32 kilodalton, then 20 kilodalton, then 25 kilodalton, then 20 kilodalton, and 15 kilodalton will have the highest migration or higher mobility. So it is P, R, P, R, Q, and S. So this is the increasing order of mobility on SDS page. Okay, so that will, but so the answer of this question would be D. For chromatography, the sequence would be SQPR, and for electrophoresis, the sequence would be PRQS. Okay. So this is the answer. This is my NPTEL reference. Thank you for watching.